Hi everyone and Salam Sejahtera. Today's topic is homeostasis. It's about a balancing act which occurs in our body. Homeostasis is a self-regulating process that regulates internal variables necessary to sustain life. In other words, homeostasis is a mechanism that maintains a stable internal environment despite the changes present in the external environment. There are three subtopics for this chapter. Firstly, concept of homeostasis. Second subtopic is negative feedback mechanism. And the last subtopic is human homeostatic organ, which is kidney. First thing first, you need to understand the concept of homeostasis and homeostatic control system. It's a system, meaning it involves few components. Secondly, the components that made up the system which involve in the negative feedback mechanism in controlling blood glucose level. Last but not least, we are going to look at the structure and function of kidney as an example of human homeostatic organ. Have you ever wondered why during a hot day your face feels hot and look red? This is a common example of homeostasis. How does the skin help in maintaining homeostasis? If the external temperature is high, the body tries to keep cool by producing sweat. Also, blood vessels near the skin surface dilate so that more heat can be lost. This helps in decreasing our body temperature. Conversely, if the external temperature is cold, the blood vessels constrict and retain body heat. Thus, this is how the skin, which is the largest organ of our body, maintains homeostasis. Homeostasis is the activity of cells throughout the body to maintain the physiological state within a narrow range that is compatible with life. Whether you dive into a swimming pool, hike in the desert, homeostasis helps cells to work efficiently, no matter what is going on outside of the body. There are many ways to define homeostasis. The term homeostasis is derived from two Greek words, homeo, which means unchanging, and stasis, which means standing. In its simplest form, it means staying the same. It is an organism's internal environment which stays the same. The internal environment refers to the condition inside an organism. What is meant by internal environment? Internal environment is all the body fluid which is not inside the body cell. Internal environment refers to the extracellular fluid which consists of interstitial fluid and blood plasma. There are many factors in the internal environment that are maintained within narrow limits. Some of these factors include body temperature, water and electrolyte concentration, pH of body fluid, blood glucose level, and blood pressure. What are the main components of homeostasis? Homeostasis involves three components: the receptor, the control center, and the effector. Each component has its function that relate with the other. The receptor receives stimulus or information on the changing environment, and the control center processes the information received by the receptor. And it is the effector that responds to the commands of the control center by enhancing or opposing the stimulus. Our body maintains homeostasis by utilizing homeostatic control systems. We already have an idea how our skin regulates our body temperature. The skin that has receptors detects changes in temperature. Either the external temperature rises or drops below the equilibrium. The control center, either the brain or spinal cord or endocrine organ, sends signal to the effector, which are blood vessels and sweat glands in our skin, to react accordingly. As the body works to maintain homeostasis, any significant deviation from the normal range will be resisted and homeostasis restored through a process called a feedback loop. Homeostasis typically involves negative feedback loops that counteract changes of various properties from their target values, which is known as set point. Unlike negative feedback loops, positive feedback loops amplify the starting signal. Positive feedback loops are usually found in processes that need to be pushed to completion, for example, parturition or giving birth. Both negative and positive feedback have the same components, which are receptor, control center, and effector, which starts from receiving stimulus that deviate from set point and makes the body returns to homeostasis. In the next learning outcome, we are going to look at an example of negative feedback, which is the regulation of blood glucose level. Homeostasis is regulated by negative feedback loops and much less frequently by positive feedback loops. Negative feedback is not bad for the body. This is a common mistake and misconception. Although the term negative is included, these feedback loops are essential and helpful to the body. In this instance, negative simply means that the feedback loops works to counter the stimulus that triggers them. Our liver plays a vital role in blood glucose homeostasis. Let's study this diagram, which shows regulation of our blood glucose level. Are you able to identify the homeostasis components involved? In the regulation of blood glucose level, which part of our body acts as a receptor, a control center, or an effector? Insulin decreases the concentration of glucose in the blood. After you eat a meal, your blood glucose level rises, triggering the secretion of insulin from beta cells in the pancreas. Insulin acts as a signal that triggers cells of the body, such as fat and muscle cells, to take up glucose for use as fuel. Insulin also causes glucose to be converted into glycogen, a storage molecule in the liver. Both processes pull sugar out of the blood, bringing blood sugar level down and returning the whole system to homeostasis, a stable state. While glucagon does the opposite. It increases the concentration of glucose in the blood. If you haven't eaten for a while, your blood glucose level falls, triggering the release of glucagon from another group of pancreatic cells, the alpha cell of islets of Langerhans. Glucagon acts on the liver, causing glycogen to be broken down into glucose and released into the bloodstream, causing blood sugar levels back to normal. What is diabetes? Diabetes happens when a person's pancreas can't make enough insulin or when cells in the body stop responding to insulin, or both. Under this condition, body cells don't take up glucose readily, so blood sugar levels remain high for a long period of time after a meal. This is for two reasons. Muscle and fat cells don't get enough glucose or fuel. This can make people feel tired and even cause muscle and fat tissues to waste away. High blood sugar causes symptoms like increased urination, thirst, and even dehydration. Over time, it can lead to more serious complications. Now, we have come to the final part for this topic. Kidney is an example of human homeostatic organ. Kidney plays an important role in urine formation, which include ultrafiltration, reabsorption, and secretion processes. Kidney also involved in the regulation of blood water content. Do you know that each individual has a pair of kidneys that consists about 1 million nephrons? Nephrons are very tiny filters, but they are capable of filtering blood and eliminating the waste material. Even with only one kidney, it can still sustain life as the nephrons are capable of enlarging and handling excess load. This is known as hypertrophy. Let us review this video about the urine formation processes begin with the flow of oxygenated blood into and deoxygenated blood out of a kidney.
Blood enters the kidney via the renal artery, which divides into smaller arteries, then arterioles. The arterioles get into contact with functional units of the kidney called the nephrons. This is where blood filtration and urine formation take place. The filtered blood then passes through a series of veins and exits the kidney via the renal vein. The urine is collected in collecting ducts and leaves the kidney via the ureters. Each kidney contains over 1 million nephrons. A nephron consists of two major parts, a glomerular capsule, or Bowman's capsule, and a long renal tubule. Renal tubules of several nephrons connect to a common collecting duct. There are 3 steps in the formation of urine: glomerular filtration, tubular reabsorption and secretion, and water conservation. Blood enters the Bowman's capsule via the afferent arteriole, passes through a ball of capillaries called the glomerulus, then leaves via the efferent arteriole. The afferent arteriole is significantly larger than the efferent arteriole, creating a blood flow with a large inlet and small outlet. As a result, the blood hydrostatic pressure in these capillaries is much higher than usual. Hydrostatic and osmotic pressures drive water and solutes from blood plasma through a filtration membrane into the capsular space of the nephron. The filtration membrane acts like a sieve, allowing only small molecules to pass through. These include water, inorganic ions, glucose, amino acids, and various metabolic wastes such as urea and creatinine, and make up the glomerular filtrate. In a healthy person, the total filtrate volume amounts to between 150 and 180 liters a day. However, only about 1% of this is excreted as urine. The remaining 99% is reabsorbed back to the blood as the filtrate flows through the long renal tubule. This is possible because the efferent arteriole, after exiting the Bowman's capsule, branches out to form a network of so-called peritubular capillaries, which surround the renal tubule. The first part of the tubule, the proximal convoluted tubule, reabsorbs about 2/3 of the filtrate. In this process, water and solutes are driven through the epithelial cells that line the tubule into the extracellular space. They are then taken up by the peritubular capillaries. Sodium reabsorption is most important as it creates osmotic pressure that drives water and electrical gradient that drives negatively charged ions. Sodium levels inside the epithelial cells are kept low thanks to the sodium-potassium pumps that constantly pump sodium ions out into the extracellular space. This creates a concentration gradient that favors sodium diffusion from tubular fluid at the same time. Tubular secretion, where additional wastes and other solutes leave the bloodstream to join the tubular fluid, also takes place. The processes of reabsorption and secretion continue in the nephron loop, the loop of Henle, and the distal convoluted tubule. These parts of the tubule also have some other important functions. The main function of the loop of Henle is to create and maintain an osmolarity gradient in the medulla that enables the collecting ducts to concentrate urine at a later stage. The ascending limb of the loop actively pumps sodium out, making the medulla salty. The descending limb is permeable to water, but much less to sodium. As the water exits the tubule by osmosis, the filtrate becomes more and more concentrated as it reaches the bottom. The ascending limb, on the other hand, is permeable to ions, but not water. As a result, the filtrate loses sodium as it goes up and becomes more diluted at the top of the loop. The medulla is in equilibrium with the loop and hence has the same salinity gradient, saltier at the bottom. Reabsorption and secretion in the distal convoluted tubule are under control of various hormones. This is how the kidneys respond to the body's needs and adjust the composition of urine accordingly. The collecting duct receives tubular fluid from several nephrons. The main function of the collecting duct is to concentrate urine and therefore conserve water. This is made possible by the osmolarity gradient generated by the loop of Henle. As it gets saltier deep in the medulla, the filtrate loses water as it flows down the collecting duct. The collecting duct is also under hormonal control, so it can adjust the amount of reabsorbed water according to the body's state of hydration. For example, when the body is dehydrated, more water is reabsorbed back to the blood, and the excreted urine is more concentrated.
Nephron is the basic structural unit of the kidney, which are packed in the cortex and medulla region. The total length of these microscopic excretory tubules is about 80 kilometers. Due to its function, it has enormous surface area for the exchange of materials. The kidney filter unwanted substances from the blood and produce urine to excrete them. A nephron consists of Bowman's capsule, a double-wall, cup-shaped swelling capsule with glomerulus within it. The lumen of Bowman's capsule is continuous with the proximal convoluted tubules lumen. The hairpin-shaped loop of Henle has descending limb and ascending limb. Bowman capsule, proximal convoluted tubule and distal convoluted tubule are located in the cortex, while loop of Henle and collecting duct are located in the medulla. Ultrafiltration, reabsorption, and secretion are the processes involved in urine formation. Different parts of the microscopic nephron involved in these processes. Ultrafiltration occurs in the glomerulus and Bowman's capsule. Blood from the renal artery flows through the afferent arteriole into the glomerulus and continue to flow through the efferent arteriole. The resulting filtrate contains waste but also substances the body needs, such as essential ions, glucose, amino acid, and smaller protein. When the filtrate exits the glomerulus, it flows into a duct in the nephron called the renal tubule. Reabsorption is a process of absorbing useful substances such as glucose, amino acid, vitamin, sodium and chloride ion, and most of the waters from the tubule back into the capillaries which wrap around the tubule. Secretion occurs in proximal convoluted tubule and mainly in the distal convoluted tubule, secrete harmful or toxic substances from the blood capillaries into the filtrate of the tubules. During ultrafiltration, high hydrostatic pressure occurs in the glomerulus due to the difference in diameter between the afferent arteriole, which is larger in diameter, compared to efferent arteriole, which is much smaller. Due to the high hydrostatic pressure, only small molecules are forced to pass through the walls of capillaries and the Bowman's capsules into the capsular space. It is the perforated walls of the capillaries and the specialized epithelial cells called podocyte form a filtration membrane which permits fluid and only small solute to pass through it. The filtrate absorbed in the glomerulus flows through the renal tubule where nutrients and water are reabsorbed into capillaries. At the same time, waste ions and hydrogen ion pass from the capillaries into the renal tubule. Reabsorption of substance back to the capillary network occurs in the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. Most reabsorption occur along the proximal convoluted tubule, involve active transport, diffusion, and osmosis. The epithelial luminal surface is covered with densely packed microvilli to increase the surface area, facilitating their reabsorptive function. The cytoplasm of the cells is densely packed with mitochondria to supply energy for active transport of sodium ions out of the proximal tubule. This will cause water to passively follow the sodium out along its concentration gradient. Along the loop of Henle, water potential gradient is created between the filtrate and the interstitial fluid in the medulla. The descending limb is highly permeable to water but impermeable to sodium chloride and other solutes. As the filtrate flow from cortex to medulla, water is drawn out by osmosis. Sodium chloride becomes more concentrated in the tubule, increasing the osmolarity of the filtrate. Filtrate concentration is highest at the bottom of the loop of Henle, while the ascending limb is impermeable to water but permeable to sodium chloride and urea. Passive transport of sodium chloride occurs in the thin segment, while active transport of sodium chloride occurs in the thick segment. This produces a high concentration of sodium chloride around the descending limb, and this will cause water in the descending limb drawn out by osmosis, creating a concentration gradient in the medulla. Water and sodium chloride moves into the vasa recta surrounding the loop of Henle. Distal convoluted tubule receives a hypotonic filtrate from the ascending limb. Distal convoluted tubule is not permeable to water but becomes permeable under hormonal control, which is antidiuretic hormone, which is known as ADH, while active reabsorption of sodium ion occurs under hormonal control, which is aldosterone. The collecting duct carries the filtrate from cortex to medulla to the renal pelvis. Permeability to water and urea is under ADH hormonal control. When the filtrate passes along the collecting duct, water moves out by osmosis to the interstitial fluid. Some urea within the collecting duct will also diffuse out along with sodium chloride, contributes to the high concentration of solute, which also lower the water potential in the interstitial fluid. This urea is recycled by diffusion into the ascending limb of loop of Henle. During secretion, some substances such as hydrogen ions and drug will be removed from the blood through the peritubular capillary network into the collecting duct. The end product of all these processes is urine, which is essentially a collection of substances that has not been reabsorbed during glomerular filtration or tubular reabsorption. Distal tubules secrete hydrogen ion and ammonia from the blood into filtrate, will help to maintain blood pH. Secretion of potassium ion occurs under aldosterone hormonal control. Countercurrent effect facilitates changes in the loop of Henle. The overall effects are the filtrate volume decreases. The output of urine is more dilute and generate a high concentration of solute in the cells and fluid of the medulla. Countercurrent multiplier function is to establish and maintain a high salt concentration in the loop of Henle extending from the cortex through the medulla. This enables water to be reabsorbed into the vasa recta. Countercurrent refers to the filtrate pass each other in opposite direction in the descending and ascending limb, while multiplier refers to the high concentration of sodium chloride as filtrate flow down the descending limb and become less concentration of sodium chloride as filtrate flows up the ascending limb. The hypothalamus produces a polypeptide hormone known as antidiuretic hormone or ADH, which is transported to and released from the posterior pituitary gland. The hypothalamus controls the mechanism of ADH secretion either by regulating blood volume or the concentration of water in the blood. ADH will increases permeability of distal convoluted tubule and collecting duct to water and urea. Maintaining a proper water balance in the body is important to avoid dehydration or overhydration. Dehydration or physiological stress can cause an increase of osmolarity, which in turn raises ADH secretion, and water will be retained, causing an increase in blood pressure. ADH travels in the bloodstream to the kidneys. Once at the kidneys, ADH changes the kidneys to become more permeable to water by temporarily inserting water channels aquaporin into the kidney tubules. Water moves out of the kidney tubules through the aquaporin, reducing urine volume. The water is reabsorbed into the capillaries, lowering blood osmolarity back to normal. As blood osmolarity decreases, a negative feedback mechanism reduces osmoreceptor activity in the hypothalamus and ADH secretion is reduced. Now, let's take a few minutes to revise all the 3 subtopics of homeostasis. You may pause this video for a few minutes, copy the URL or scan the QR code. Good luck!